That now. Do we think that's working?
Yeah, I think it's working. I got a message that it was recording.
Perfect. So I'll stop sharing and hand over to Mariana. Thanks, Mariana.
I'm not allowed to screen share. Would you give me permission?
Yes, I'll do that now. Or Quentin will do that, depending on which of us can. Are you able to do that, Quentin?
I'm looking into it.
I think now I can
Oh yeah, I see you now. Okay. I will make you a co-host. Here you go.
I'm there.
Great. Go for it.
Okay. Yeah, well, thank you for the invitation. I'm gonna tell you about one of my favorite projects that's running in the lab right now. That's the monitor, and there's this automated moth monitoring done by undergraduate students. And yeah, it's the insect declines what got me to this project, which is really outside of my comfort zone. Well, not anymore. So this was funded by SEEDS, the Center for the Environment, Ecological Design and Sustainability here at Smith College. And we have two types of goals. One set is regarding monitoring. So we want to establish a local moth monitoring program. We have a field station here, so we want to establish that first. And once that's running smoothly, it's not quite there yet, we will expand it to include multiple locations in Massachusetts. And once we have the logistics solved for coordinating multiple locations, we want to expand it internationally to include this tropical dry forest in the west coast of Mexico. The reason I'm interested to expand there is because it has interesting seasonality driven by rainfall. But at the same time that we have the monitoring goals, there are some goals for education. So I want to foster insect appreciation, interdisciplinary collaboration that's more and more important in this recent world, and also appreciation for the dark sky and to fight light pollution, which is a pretty ambitious goal, but we need to get there. And this is also a very good opportunity to reach students that are outside the fields of biology and environmental science. So those that are not already interested in insects or worried about the insect declines. So we started with the first monitor in the summer of 2022 with my students, Ray and Kate. And so they went to the Center for Design and Fabrication. I'm going to mention a bunch of students that are working here, and their names have the year when they graduated and when they will graduate after their names, and then mentors don't have years. So they went to the Center for Design and Fabrication, and they made the frame for the monitor that's here. So it has a metal frame with a wooden and fabric landing surface. And it works with a waterproof camera that you can just buy. It's waterproof and it's programmable. Then it has a UV light that has a custom-made case to make it waterproof. And it can be plugged to a standard USA outlet, which uses an extension cord, and then this mechanical timer to control when the light goes on and off. And then some metal stakes for field installation. And with that setup, we got this kind of images. They're kind of blurry, so we don't have very good taxonomic resolution. We could identify moths, mosquitoes, and insects in general. So this would be the equivalent of monitoring grams of insects, like biomass, but the non-lethal equivalent to that. But then once you get the images, you need the software to identify the insects there and label them, count them. And for that, we had the computer scientists join here, Emma and her professor, Nick. They work on this as part of a class project that happened last year, computer vision and image processing. And what Emma did was a Python program that uses a neural network that's based on AlexNet. And she got 81% ID accuracy to the three taxonomic categories that we have, mosquito, moth, or insect. So the Monitor 1.0 was easy to assemble. It can estimate insect abundance, so just count, but it had low taxonomic resolution. It has restricted scheduling capabilities because it's all about what you can program in the camera, and it's not a lot you can do. You can take a picture every 90 minutes, pretty much that's it. And then it had a restricted location. It had to be close to an electric outlet, so it was close to a building. And yeah, we got some data from that. And as I said, you can get abundance and counts, but we wanted better images. So this is when the engineers joined the team. So Melanie, Margaret, and Millie with their mentor, Susanna, and they, and also Ray stayed in this team. And we split the team in two groups, one that was working with a wildlife camera and one that was working with a Raspberry Pi controller. And this is the Monitor 2.0. This was developed last spring, and it's very similar to the first one in the sense that it has this frame, but this time it's aluminum, so it's way lighter. And it has a hunting trail camera that's better for programming, but it doesn't have a flash. So if you want lights, you need extra lights, and that's why we have this strip light with a digital timer. And this time we have an outdoor solar powered battery, so it's better for portability. And this is the type of images that we got from that. So it's definitely better image quality. The taxonomic resolution would be better, much better programming capabilities, and the portability, but it was very low energy efficiency. And as I said, at the same time as we were working with this wildlife camera, the other engineering team was working on a Raspberry Pi controller. But while working with that project, we realized we need more computer scientists. So that's when Heather and Pablo joined the team, and they had experience working with Raspberry Pis. I don't know if you've worked with those, but they can be, they're advertised with these fruits and they look colorful, but that doesn't mean you can learn to use them in a week. You really need someone who knows their way with those. And so Heather and Pablo knew their way with that. And so here you can see the setup in the field. Here's a schematic, so it's easier to understand. So we have the controller, the Raspberry Pi here, that's connected to a camera. And then the controller is connected to a relay that controls the light that works as a flash. And then the battery is also connected to the light, that the UV light that attracts moths. And this is the camera. This is the lamp that acts as a flash. And this is the relay. To make a relay, you really need someone who knows their electronics. This is not something you send your students to do on their own. And this is the type of images that we got. So much better image quality. I was very excited when I first saw these pictures, but if you know how computers think, you can see that there are some bolts here that can be confusing, and then this shade. So we ran Emma's program to identify the insects, and as you can see, it can identify a bunch of moths. There are some that are not being detected, and then the bolts are there. But now that we have much better image quality, we have to label the moths, so identify the moths, and that's a lot more work than last time. So now we have a labeling team, Lainey, Ray, Sabrina, and I. We've been working this semester as part of a special studies class in which we learn about moths, about their ecology, their taxonomy, and then they help me label the images. So the current status is we're still labeling them. We hope to be done with that by the end of this year. So Emma's program can be retrained next spring. So we definitely had much better image quality, but the labeling process was slower. The landing surface has some irregularities that interfere with moth recognition. So we have a team that's working on the Monitor 4.0 that we hope to launch next summer. And so we have Heather and Pablo that stay in the project, and Claire, who's an engineer who just joined the project. And we want a waterproof case for all the electronics. You probably noticed they are in deli containers. So we want them to have their proper waterproof case and a larger and more uniform landing surface with a scale. So as you can see, this project involves 5 mentors, counting me there, and 10 students, and we hope that it will keep growing and getting better. And with each generation of students, the project improves, and we also reach more and more people with the message of insect conservation and conscious about light pollution. And with that, well, I want to thank you for your attention. I want to thank Smith College and Seeds because they truly create an environment in which we can foster this type of collaborations among people from multiple disciplines. And of course, Wildlife's net, because having a network where you can go and see what people are doing and chat there, that has been very helpful for this project. Thank you.
Thanks very much, Mariana. I think, yeah, it's really nice to see the story and all the different stages. And I think what's unique about the project is how it seems like it's really kind of driven and pushed forward by the students, by a big group of students, which I think is really super interesting. We have budgeted a few minutes here for questions, but just so you know, there will also be a session at the end to have a kind of a panel discussion around this stuff as well. So don't feel like if you don't get your question addressed here, that you won't have a chance later. So yeah, any questions for Mariana at this stage? Dennis, would you like to ask your question?
Yes. First of all, thanks for your talk. I really enjoyed it. And I have one question to your approach with the AudioCam. When you set up this approach, have you tested the resolution of this AudioCam? So I know it's labeled with 64 megapixels, but do you know if you really zoom into the picture, if it also gets blurry? Because I'm also working with such a device now, and we tried to measure the resolution of it, and it was not so nice.
Yeah, so what we did, well, what the computer scientists did for that is when the picture is taken, is not just taken, but the camera adjusts, like gets the image into focus. There's some way in which you can program it to do that. And once it's in focus, it takes the picture. And we also take 2 sets of pictures with different, what is this, like the aperture, like with different lighting. So one is brighter than the other. But what's challenging there, once you have, it works in the lab when we have dead moths that we tested with, but then with the live ones, you can have some that are larger than others. So getting everyone in focus in the same picture is hard. So I would recommend taking multiple pictures and programming that step of getting the image in focus before taking the picture.
Yeah, basically my question was not about having the focus, but we tested them with standardized charts that you could use for TV cameras, where you know how many line pairs per height are resolved, and there it was definitely on focus, but the resolution was quite low. That's the reason why I got thinking about it.
And did you get the 64 megapixel camera?
It was a 64 megapixel camera, yeah, and the resolution was at the end quite low.
Oh, yeah.
Lower than compared to a 24 megapixel camera, so.
Oh, no, then there's something going on there, because yeah, we can zoom a lot in our images, and well, we can save some that are very high resolution and then lower the resolution so they don't take as much space. But yeah, I'm not sure how to fix that. Maybe the camera's not working well.
Andy's saying in the chat that there was potentially some up-resing going on here. Is that the case, Andy?
Just on that topic, so I've also been looking at these over the past year, and it seemed like there was a bunch of weird stuff going on with AudioCam and this particular camera, where some people in the forums were like, hey, you're not actually giving a 64 megapixel image, you're like up-resing a 16 megapixel image. And there was like the drivers were like a real hot mess for a little bit, but at least as of like this past summer, they seem to have Northern Hemisphere summer, they seem to have stabilized a bit, at least for me. So like I feel like I've seen kind of what you're talking about, where it was like I was just getting garbage from this thing, but now it seems like I can get better full resolution, hopefully, images. But yeah, I kind of know what you're talking about, Dennis.
Okay, thanks. Then I will discuss this with our technician and see if we can work on this. Thanks a lot.
Yeah, good luck, Dennis. So let's move on now to Jacob. If that's okay, Jacob, if you're ready. So Jacob's from the University of Florida, and he's going to talk to us about the AutoMoth system, which also comes, I believe, with some software toolkit that people could potentially engage with. So I think you've stopped sharing, haven't you, Mariana? Are you ready to share, Jacob?
Yeah, sure.
Great. And Quentin, can you do the thing if, yeah, great.
All right, yeah, is that visible?
All visible, take it away.
Great, yeah, so I'm Jacob Idek. I'm a third-year PhD student at the University of Florida, advised by Rob Gorolnik, and I'm going to be talking about the BioLens and AutoMoth Community Science Toolkit for Automated Nocturnal Insect Monitoring. So the goal of this project really is to scale up our ability to monitor. We want to increase the spatial scale of monitoring in terms of monitoring in more locations, and the temporal scale of monitoring in terms of monitoring more days during the year and more times during the day. And, given the success of platforms like iNaturals in generating huge amounts of biodiversity data, we think we can leverage this kind of community passion to get people to deploy, hundreds of thousands of these automated monitoring devices. So we're really wondering how we can facilitate a network like this. So what you want to cover is BioLens, which is a free mobile app for biological interval imaging, which was designed with monitoring in mind specifically. It basically lets you use an Android phone as a complete camera module. So it's a camera module in the sense that you can configure it to take images in a certain imaging interval, taking them at certain times. It also records and attaches associated metadata, things like geolocation and time. And it can also do some other neat things like allowing you to share images easily or upload them automatically to the cloud. We're also working on our own low-cost automated trap design. We want to allow more people to deploy more devices. So yeah, I'm going to jump right into how BioLens works and outline some of its design. On the bottom right is Jean Hostetler, who did all the programming for this app and also contributed greatly to our thinking and design around this stuff. So the main functionality of BioLens is setting up sessions to run at specific times. So on the left, you can see we're setting the imaging interval. This is how long to wait between an image. We're setting it to stop after 12 hours of imaging. We then select a date and time to start the session and give it a name and hit schedule. The phone will then, as long as the phone is turned on, the app doesn't need to be open though. It will start imaging, record those images, record that metadata. So yeah, if you wanted to set out this module for a week, you create 7 sessions at the times that you want. And it would do the imaging for you. So one of the really key features of BioLens, something that we think about a lot, is maintaining metadata for the images. We expect a lot of variation across imaging setups in things like the model of the phone used, the size of the sheet, what lights are used and how they're positioned. And it's really important to keep track of that data if we want to analyze a lot of data from a lot of different kinds of imaging setups in tandem. So we store this metadata as a CSV alongside the images. Some of the default fields are the sheet dimensions, whether it has a scale bar and color standard, the models of the UV and white light, and the model of the smartphone. It sort of the two most important pieces of metadata are the geolocation and the time, and that's automatic, which is kind of nice. You can also attach custom metadata, whatever fields you want with whatever values, depending on maybe your specific situation. And on the left, you can see an imaging session. Within the app, you can do things like browse images in a session, upload a session to the cloud after the fact, and also some neat things like cropping images to individuals for upload to the iNaturalist app for identification. Something that a citizen scientist very well might want to do if they're interested in a certain individual. So yeah, this is our settings panel talking. So BioLens supports either immediate upload, so uploading images as they're taken, or upload after the fact, currently to Google Drive, although we want to expand this to different kinds of image hosting platforms. Maybe, hopefully, I think eventually our own platform for hosting images from different devices. And this can use either cellular internet or Wi-Fi. And it can be really nice to sit back at home while this thing is plugging away, uploading images for you. You can see them rolling in, at least in situations where your device is deployed in a place with internet. So yeah, no one on our BioLens team has been a hardware engineer for some time. So our initial hardware for AutoMoth, I would say, was like pretty messy, kind of ugly. But recently we took on Charles Bradley, who Rob kind of randomly met, who is strong on hardware development and actually had already thought a lot about how to design blacklighting setups. So that was really convenient. So yeah, our hardware framing is mostly PVC. It has a mount that you can attach a phone using BioLens to, a regular camera, or a Raspberry Pi based module. It includes a white ring light and a UV bar light to do illumination and attraction respectively. And we're aiming for a total cost less than $100, which it helps a lot when you're buying these components sort of from scratch and putting them together. So this is what that looks like. Yeah, you can see the illuminating white ring light, the UV bar light, and the camera mount as well. So we're still working on and testing final specs for our device. The goal eventually is to scale up production and produce specs that people in the community can use to build their own, and also eventually build and sell units for people is the hope. So some results from an AutoMoth test in Tucson, Arizona. Note that these images are downscaled. If you look up closely in the final images, I would say it maintains detail in medium to large size moths, probably not the micros, but it's pretty good with medium to large ones. And of course, this varies based on the phone model you use. So yeah, some quick thoughts on computer vision. I know Mariana hit on some of this stuff. I do think, as her computer science students showed, insights can be extracted relatively easily from light turb images. If either using non-machine learning approaches or more sophisticated machine learning approaches, I think foundational models like segment anything would be really effective on a data set like this. This gives us a series of segments, each one being one insect at one period of time. For each segment, we can get information like a species ID, either by experts or automatically using machine learning. I think eventually it'd be amazing to use iNaturalist's machine learning models that they've trained to identify species in data like this. We can also get, if we know the sheet size, we can get measurements of body area, perhaps eventually the size of specific body parts, and also other traits like mean body lightness, which is an interesting trait for thermoregulation and other important ecological functions. And using the segment data, we can get some really cool metrics, things like the number of individuals or species on the sheet, a proxy for biomass, the number of observations of a certain species, a proxy for abundance, and also some really cool trait data. I think if we were to scale up this kind of data, we could do some really cool studies on how body size or color varies within species, perhaps across the species range, or depending on the climatic conditions in which they live. So a lot of possibilities for this data. So some more gaps that we're thinking about. We think it's really important that we standardize metadata across camera traps as best we can, and people using camera traps should try to maintain their metadata as best as they can. And it would be really, really awesome. This is sort of maybe a lofty goal, but we think one that is attainable eventually. Building a place for images to be from both BioLens and any imaging device to be stored in the cloud, shared in the cloud, and also perhaps even analyzed on the cloud. So we can get metrics. You could upload an image and then get metrics for it, or download those metrics even rather than the images. Things like use cases like that would be really cool, I think. And I can imagine this facilitating citizen science as well. It'd be really cool for citizen scientists to be able to upload their sessions and having things like abundances, IDs, trade information come right back to their devices. That would be a really sweet deal, I think. And yeah, once again, our goal is to lower barriers for community scientists and provide a low-cost camera module option. And yeah, we see Automoth and BioLens as tools to help us start creating large-scale monitoring networks, particularly community science ones. One more story. So while we designed BioLens to focus on moth monitoring, someone in our lab actually used it for a big experiment on thermal choice and disease dynamics. So we have these salamanders here in these boxed enclosures, and a thermal strip sort of causes variation in temperature across these enclosures. And then we're using BioLens to track the positions of these salamanders, how they're moving across the enclosures, to either to modulate their body temperatures, depending on whether or not they're infected with chytrid fungus. And the experiment has started to yield some really cool results about how thermal ecology interacts with disease dynamics. So yeah, sometimes tools can have value more broadly in interesting ways. We thought that was neat. Yeah, and you can download BioLens now if you want. It's available on Android. And yeah, if you do, please give us feedback. Yeah, we love to hear from you guys. Yeah, thank you.
Thanks very much, Jacob. And yeah, it's not always pretty, but what I think is great about your solution is that it's really ready to go, and people can get this app and they can start using their phones to do something that other people might be maybe you don't always need a Raspberry Pi to get this stuff done. And I think that you've shown that really well, which is super. And I hope people will try this app and give feedback. Any questions for Jacob? We have a couple of minutes before we need to change over to the next speaker. Quentin, your hand is up.
Yes, thank you, Jamie, and thanks, Jacob. Really nice talk. You mentioned, I also really like the pragmatism of your solution. You mentioned, of course, supporting various devices, and I'm wondering if you have a specific strategy for the future. When Like, I'm assuming this will be for dozens of different camera specifications, and this will change maybe in the future as if camera get better and so on. So how do you anticipate standardizing the image quality itself, if at all? Thanks.
Yeah, so we definitely anticipate, if this catches on in community science as we're hoping, there'll be a really broad range of cameras that are used, and also types of hardware that are used. And I think, to me, like we'll have, of course, suggestions, like suggested specs, suggested phones, but regardless of that, there'll be a lot of variation. And I guess in my mind, the best way to sort of, I think the key with that kind of variation will be sophisticated metadata. I think if we can keep track of the specifications of these variable devices, and the lighting conditions, things like that, it'll go a long way in allowing us to use images from a variety of different setups.
Thanks.
Hey, Jacob. Hey guys, this is Rob. I can also jump in and say that Jacob got the key answer. I think metadata is really important here. We've thought about this through the lens of like getting the cheapest possible high quality cameras that you can purchase. And so like we've actually checked the market pretty carefully for like higher quality camera phones that are like a generation behind where we are now. Our feeling is, and this is up for discussion, like there's lots of movement across Raspberry Pi based cameras or DSLRs that are cheaper and really lightweight and usable in the field. They have different cost points and different values, but like one thing that we're wondering about is whether, like, as we see generational shifts in camera quality, whether those previous generations are going to be so good that it's sort of maybe looking at a nice set of trade-offs, right? Not the set of trade-offs we have now, where it's really hard to figure out like the best possible solution for the lowest possible cost. But I think our solution is meant to basically leverage previous generation camera phones that are good enough to use in the field. There's a whole bunch of like weatherproofing and field proofing that we can talk more about that we thought about, but I think it's a really interesting question.
Yeah, and you can imagine a scheme where people were able to donate their old phone, which is basically valueless to them, for biodiversity monitoring initiatives. I think like it's something that you can see happening, but we'll see. Let's move on to our next speaker, who is Andy Quitmeyer. And I believe, Andy, you're in Panama, based at Digital Naturalism Laboratories. Hopefully you can tell us more about that. And I'm going to need you to unshare your screen, Jacob.
Yeah, so howdy. I'm coming at you all from Panama right now. Anyone else here currently in Panama? I know there's a big moth crew that's just right next door. We're in Gamboa here. We got a big science island called BCI right in the canal off next to us. But here, let me share my screen. Cool. Can you see my screen?
We can see your screen. And we can see the different slides on the left. It's perfect, full screen now.
Okay.
Or
is
it?
It shared to the wrong side. Monitor. Go to this one, please. There we go.
That's perfect.
Now you're seeing pictures, right? Here's what Panama looks like. This is right next to us. It's beautiful. There's all kinds of nice little mist and stuff like that. It's a shame the government's trying to push through a giant evil death mine to destroy the whole biological corridor of the Americas. But more on that later. I just want to say, hey, I'm at Digital Naturalism Laboratories. We're right here in Gamboa, Panama. I'm Andy. I founded this little tiny anarchist maker space with my partner Kitty here. We call ourselves Dynalab. There's lots of goodies out here that are buds. And our main things that we do is we create art and technology for interacting with nature. And we try to do this as much as possible in the wild or close to the wild or next to things, so we can get lots of feedback. When you're designing things, it's very useful to get rich feedback. And so I'm right here, just in our little house maker space. It's a solar powered place with little workshops and facilities, electronics and laser cutters and 3D printers. And we also do our own upcycling. So about 90% of our prototyping material that we build stuff with, like when we were building this little moth box here, most of the material is all garbage, upcycled plastic or paper. This is an Olingo GPS collar we printed from a plastic bottle. We run some other events called Dynacon. We do those around the different world, interacting with different communities. But one of the big things that we do is we try to help do designing and consulting with field biologists. So I have a computer vision background. And one thing I learned early on when I was doing my PhD is you can make all this great stuff for looking at bugs or whatever. It works amazing in your nice climate controlled laboratory. And of course, once you bring it to the wild, the first thing it does is break in all kinds of wonderful different ways you didn't even anticipate. And that's kind of the beautiful part for me. And so that's why I try to move into this kind of contextual crafting. That's why we have a lab here right next to where we want to test stuff in Panama. And so there's lots of different projects we work on. We make 360 camera traps. So that's if you were like a regular camera trap. Oh, we got one agouti. But 360 camera trap, what? There's 4 agoutis right there, right? Other examples, just random things like a butterfly strength tester. These are floating fishing bat camera traps that stabilize for the waves and stuff that use the same nice programmable waterproof camera that we saw earlier with the first presentation. Nearsighted camera traps, hacking regular camera traps to study butterflies, even though they're close and tiny. And things like the moth box that I'll talk about in just a second. And everything that we do is open source because if it's not open, it's not actually truly science. You need other people to be able to recreate things, to learn from things, to share, to build on each other. So make sure to open up your stuff. We also teach field courses, build infrastructure. These are boat labs that we built in kind of community maker spaces in the Philippines and Sri Lanka, teach design courses. We do field maintenance and do community building, kind of Spanish and English classes around here, do long and short-term residencies. That's kind of Dynalab a little bit. But let's dig into what's this little thing that we're making here. It's a fun glowing thing. It's got little rods. It's pretty cool. Guess what? It's used to study moths. That's the big spoiler. Another fun fact, when we were creating it, Hubert, the kind of instigator of all this, was like, hey, let's make this. I'm like, what should we call it? We're like, oh, it does like automated moth detection. Maybe we should call it the auto moth. But then we found out y'all had already beat us to the cool name. So then I was like, I guess we'll just call it like Mothbox for now and come up with a better name later. So I present to you Mothbox, the name that sticks. And so it's a low-cost platform to capture high-quality images to try to identify insects. And so the background, where does this kind of start? This is Panama right here. And if you've been to Panama, it's super beautiful. There's lots of different ecological niches and stuff. It's a really fun, neat place. And there's an area right here called the Azuero Peninsula. I don't know if you can see my mouse or not, but I'm circling this kind of thing sticking out the bottom. And around World War II, the Americans were there. And they were like, you know what? I'm in this cool tropical place, but I really want hamburgers. So I'm going to tell the whole local population to go chop down all the rainforest there and actually grow cattle farms. So there's a ton of deforestation because of cattle farming. And when you actually visit there, when I first visited there, I was like, oh, this is just a different area, a climate of, Panama. This is kind of like a dry, deserty California feeling area. But no, it was actually used to be rainforest just like 80 years ago. And so there's a really big, cool initiative called Azuero Reforesta. And they are working with the community there to help try to get philanthropic money into the hands of farmers and local community members to actually maintain forests they already have, rejuvenate forests that have been destroyed and turned into farms, and also pay farmers to cultivate native seedlings. So this is different than a lot of reforestation projects. Apparently, this is a shady thing. Only like 8% of reforestation projects around the world actually use native trees. Other places are just like, we put trees in the ground. We grew a giant monoculture of teak. That's fine. But so they're actually trying to do this right, and it's really impressive. And one of the other ways that they're doing it really cool is there's a bunch of scientists like Hubert Stigl, who is a PhD student helping monitor and study what's happening with this reforestation project. So, hey, we're going to try to change the landscape. What's going to happen? How does this affect? How effective is it? Their team is actually doing a lot of really cool things. They're not just studying the biodiversity impact, but they're studying like the economic impact, the cultural impact, people's view of nature before and after kind of these transformations. But Hubert's studying kind of a broad spectrum biodiversity project. They're doing like traditional methods and kind of newer approaches like passive acoustic monitoring, but also part of his work is about making fun new things and testing them out. And so this is kind of like what we're trying to do here with the moth box to try to help standardize monitoring local moth populations. And so we've been working at this since December of last year, so a little less than a year. And it's gone through a couple different phases, from just, a bunch of stuff in a Tupperware jar to hopefully getting a little more refined and portable. It started off with a little bit of tiny funding from Earthshot to help develop this thing. Then a cool group at Michigan State University kind of took it up and tossed in a bit more money to help develop a version 2. And now we recently got about 10K from experiment.com to kind of develop this more in Panama and also the US and different places around. And then Hubert actually got a little bit more funding to make 10 of these that he's going to deploy in January. So it's a very scrappy little project that's, we're just getting through on what we have, but it's pretty fun. And so some of the functional targets is that Hubert basically has to go carry these things very far to like very kind of remote areas that he's been told he can only access maybe once a month because it's going to take a lot of time to like, trucks to 4-wheelers to hiking to like set these up. And so it's totally unmonitored, off grid. We're not connected to anything. And his goal is to kind of have at least 5 nights that he can do a kind of photo capture on and try to turn lights on and see what's there on an A4 size target. You can see this little guy sticking out there. And also another target goal of this is it needs to be easy to train non-experts in how to use this. And so it'll be running out there, hopefully be able to identify things down to the genus or species, hopefully. It's got the 64 megapixel Ardu cam. We got a little power manager to try to put it in low power mode. And it has built-in little LEDs for lighting up and camera capturing, as well as one thing that we like is it's kind of more of a platform. So we made waterproofed USB connectors that we can plug in all kinds of different types of lights. And more on that in kind of a second. But one of the other things that I really wanted when I was making this is it's all open source, and I wanted it to be very easy to build and with just using off-the-shelf parts, minimal wiring electronics. And importantly for me, everything's laser cutterable. And that's because if you've done like 3D design stuff before, it's a bit harder to make everything out of only 2-dimensional sheets in a 3-dimensional thing. But it means that we can whip out things much quicker and cheaper. And if we don't have a laser cutter, we can always print them and then jigsaw out different parts like that. Or if we have a 3D printer, not a laser cutter, we've done this before, we could 3D print the different shapes and things like that. It has different modular stands. You can attach any kind of tripod or thing like that. The images are 9,000 by 6,000. The whole thing's about $400. We can get currently on the battery that we have in it about 10 hours of UV photo capturing time. So we did 5 nights. We get about 2 hours each night over the course of a month. Of course, we can change this around. There's some things we're looking into to make it a little more power efficient, but that's currently what we got with its little integrated battery. Other things that I really like about this thing is thinking of it also as a platform. And this is kind of my last little thing that I was thinking about is just for any kind of people maybe from the engineering side. You know, when Hubert's first like, hey, let's build this thing to detect moths, I'm like, great, here's all my list of questions. Like, what types of light do moths like? What types of surface do they like to go onto? And, you know, from the engineering side, you're expecting like, oh, this is going to be this exact answer. They prefer 365 nanometers and they want this. But it's actually like, I don't know. This guy says this. This other person says this. And so then I'm like, what? But we're kind of designing it modularly. So like, you know, we can have different targets with different, you know, woven material, felt material. It's yellow, it's white, who knows? But the nice thing about a little platform like this is that we can set up like 4 or 5 of them and, you know, see how they work. So we're going to build 10 of these in December. Hubert's going to deploy them in January, and we'll have all the documentation up and shareable on as many platforms as we can so people can quickly build them themselves. Yeah. And we found a little buddy that's been some sort of undescribed mantis while we already did initial testings in the Azuero Peninsula. So yeah, so that's me. I'm Andy. Feel free. Any questions or anything like that?
Thanks, Andy. So you used up your individual question time. We're going straight into a discussion anyway, and it was definitely worth it because these pictures are absolutely stunning. Yeah, yours is definitely the most compact design I've seen, and I think it's just great to see how different all these different solutions are, but ultimately they are working towards the same thing. And so hopefully in the discussion we can talk more about how we, you know, allow for that diversity while also generating useful data from all these angles. And then hopefully Jenna's ready to give us a quick run through of the AMI system, which is, to my knowledge, the most widely deployed automated moth system so far. Yeah, are you ready, Jenna?
Yes, I am. Bear with me 1 second. I'm just going to share my screen.
Brilliant. So Jenna's from the UK Centre for Ecology and Hydrology.
Okay.
Yeah, so we call it AMI, standing for Automated Monitoring of Insects. It's another case of name being occupied.
Can everyone see this?
Yes, looks great. Far away.
Let me try and minimize this. Okay, hi everyone. So yeah, I'm from the UK Centre for Ecology and Hydrology, and I'm just going to give you a quick rundown of the AMI system, which stands for Automated Monitoring of Insects. So the first thing I just wanted to go over is sort of what is the gold standard of data that we're looking for? And I just wanted to go over this because I think that this is what we're all working towards with systems like this. So generally, when we collect ecological data, we're looking for data that's non-invasive or less invasive. And new technologies are definitely helping with that. We're looking for data to be non-biased or as least biased as possible. We'd like data at wide spatial and temporal scales, high resolution. We want it to be standardized. We don't want people collecting data in 1,000 different ways and then it's not comparable. We'd like our data to be verifiable so we can actually test it. And then we want it to be balanced, not too much versus not enough. And I think that the systems that we're developing are going some way to addressing this gold standard of data that we're really looking for. And so this was called the AMI trap. We've started calling it the AMI system because people thought we were killing things by calling it a trap. So the AMI system, you can see a picture of it here on the right-hand side. So it consists of a whiteboard with a LEPI-LED UV light on the top, and then a motion-detected camera with a string of white lights around it. And so the lights are attracting the insects onto the board, and then the camera is motion detected, so it takes pictures meters every time it detects motion on the board. And then it also takes snapshots, and you can take those snapshots at whatever intervals you'd like. And you can also turn the motion off and just have snapshots if you'd like to. And then all of this data is then stored on currently a 500 gigabyte hard drive in the back, but you can put 2 terabytes, 5 terabytes in there, anything that you'd like. So there is high capacity data storage. It can be plugged into the mains power, or it can be powered, as you can see here. We've got this is out in a field in the UK. It can be powered by solar panels. The sampling schedules are customizable, so you can record on any schedule you want, different days, different times of the day. And then also there is, we use an AI system which was developed by the Rolnick lab in the US to detect, track and identify the moths. And this is just, hopefully this will play. This is an example of the moth trap out in Panama last year. So you can see it's a very busy system. There are it has undergone some developments since then. This system's actually, it was developed quite a few years ago by a group in Denmark, and it's been developed further in collaboration with that group and other groups in Europe, the US and Canada over the last few years. So it's not just the UK Centre for Ecology and Hydrology that's been developing this. And this is just an example of the AI system developed by the Rolnick lab. So it's a 3-stage process. It first uses object detection to detect an object on the whiteboard, and then it uses a sort of a non-binary moth or non-moth, and then it goes down to species level for the moths, and it gives you an accuracy of all the different detections. And this AI classifier is currently pretty good for Europe, the US, Canada, so for more northern temperate regions, and we're working on tropical regions at the moment. There should be a map here, but it seems like the map's been cut off. So where are the Amy's? My lovely map was going to show you, but I'll just have to tell you. So we have well over 100 of these systems at the moment across Europe, the USA, Canada. We've just deployed 6 in Panama this week, and in Argentina. And then we're currently working on a very large scale project called Amber. And this is deploying and testing these units across Panama, Costa Rica, Singapore, Japan, Thailand, and Africa. So we've got about 50 units going out over the next 6 to 9 months in these countries. And the idea behind this is to test out these systems in tropical ecosystems, and also to be able to collect the images to develop the AI for all of these understudied hotspots. And so this is more to sort of kickstart the conversation about some of the benefits and challenges of these ecosystems, these systems, and where we see the development of these systems going. So the benefits and the usefulness of these systems is that you do have wide temporal monitoring. So these systems can stay out for months at a time, as long as nothing breaks on them because of their use of solar power. The data collection is standardized. We believe that there's reduced bias in the data that's collected. It's obviously less invasive because we're not going out and trapping and killing moths. The data is quite high resolution. It's verifiable because we collect and save all of the data. It should be cost effective because even though there's a cost to the original devices, it does reduce the person power that you need in the field. But on the back of these benefits, there's obviously challenges. So at the moment, we're struggling with a power supply under closed canopy. Often we want to place these devices away from mains power. But when we do that, if we're under a canopy, we're relying on solar panels at the moment, and we're really struggling to get sort of a lot of power under these canopies. So we are actually looking for other power sources at the moment. Hardware and software development is a constant challenge. At the moment, just deploying these devices in Panama this week, we've noticed that there is a little bit of issue with humidity and the high rainfall, so there needs to be further modifications to the system, and the software for this is constantly being developed as well. There is some bias, even though we do reduce the bias because we're not present in the field and killing the species. There is bias from the light source that's introduced, which needs to be minimized as much as possible. Equipment can malfunction, and often it does, especially in the tropics. Data collection and storage is an issue. We are collecting large amounts of data and we need to find some way to store that. And then also processing of big data is bringing challenges across every new form of tech that we're deploying at the moment. And so just some of the future developments that we're working on at the moment to help solve some of these challenges. We're adding telemetry to our system so that data can be sent across the network. So rather than it having to be stored on the actual device. And that also brings the ability to do remote health checks. So hopefully within the next couple of months, we'll have remote health checks up and running, and that will allow the system to tell you when it's broken. So rather than thinking that you can deploy for 3 months at a time and then coming back 3 months later and finding it broke on day 2, the system will actually tell you that it broke on day 2. We are adding on audible and ultrasonic acoustics to the device because we figured if we're deploying in these locations, we should be maximizing the amount of species and taxa that we can study. And we're also working on something called the Amy Mini. This device is quite expensive right now, and it's large and it's difficult to deploy in remote locations. So we're working on a mini system which will have probably reduced temporal scale of monitoring, but will hopefully increase the spatial scale, and will probably be used by different researchers to answer different questions. And then we're also working on alternative power sources for when we're working under closed canopies. And some of the software that we're working on right now, we're building data pipelines on the cloud so that all the analysis, the data can be uploaded to a cloud and all the analysis is done on cloud. We are developing edge processing in combination with the Turing Institute. So this is where all the actual analysis will be done on board, and then the only thing that's sent back across the network would be the actual results. We are doing a lot of work at the moment to compare manual versus automated methods to see how these methods with new technology are actually comparing to standardized moth monitoring. Are we missing certain species, for example? And then we're also looking at identifying and quantifying some of the biases that we're seeing. So, for example, is there an issue for bats by introducing a light source into the system? Currently, we're finding not, but there's a lot more research to do around that. And then the classifiers are continually being developed and improved. And so that's it for the Amy system, and hopefully that kickstarts the conversation, the panel discussion.
Yeah, it definitely does. And yeah, I think you covered a lot of the big challenges there. And I think the Amy system is forming a good platform in Europe to investigate those challenges. I think so the plan is we want people to try and submit their questions in the chat, although hands up will also be is also accepted. And then yeah, we're gonna give people we're gonna treat the speakers today like a panel, and yeah, hopefully cover a few things. And while you guys think of the kinds of questions you want to address, I might just throw out the first question as basically what I think is really interesting about this is that we have different valid technologies for the same to address the same problem, which is essentially monitoring moths and maybe nocturnal insects more widely. And I guess I'm coming into this with the angle that coexistence of these different approaches is definitely a good idea. And Jenna made a really good point that it's better not to have people collecting data in 1,000 different ways. But maybe I'm wondering if the number is less than 10 different ways, then we can maybe we can, as Jacob said, capture the metadata appropriately, that we can account for these things later on. So I might just put that. I'm going to say Jacob first. Have you got any thoughts about that subject of fitting in all these different technologies while also generating data that are sufficiently standardized?
Yeah, it's a tough one, I'd say, because like you know, like I mentioned before, you know, I think we can provide, so, and also Robin has some thoughts on this as well, but, you know, I think we can provide, I think with regard to citizen science, we can provide suggestions to people, but we can't expect people to always follow them. You know, like much like iNaturalist, it's kind of incidental data. And I think with citizen science, at least, we should anticipate variation, you know, ahead of time and try to account for it in the metadata. But I think as scientists building networks, I think, you know, we do have the possibility to try to standardize our metadata fields more explicitly, and I think that would be really useful.
I'll back that up, Jacob. I think maybe less important than heterogeneity on the hardware side and different implementations that fit different use cases. I think everyone's going to design towards a set of use cases that they have. Ours was what's the cheapest possible design that's light and portable, field ready, that can be deployed, you know, really effectively. Our main audience is to bring these to nature centers and other places where we can have these deployed across a gradient, large spatial gradient, and build these out with a minimum of cost and a minimum of challenge with people setting them up and using them. That was really the goal of what we wanted to accomplish. And so I think everyone has just different goals on the hardware side, and it's not going to be possible to standardize that. But I think we definitely need to work really hard at trying to build a better way forward on the ability to share these data and also build out the metadata frameworks that are shared so that this doesn't become a hodgepodge of data that we can never recombine.
So Quentin's got his hand up, and if anyone wants to jump in at any point, please stick your hand up, especially the panelists.
Thanks, Jamie. Yeah, it's what Robert said makes sense, and it's kind of bouncing back to maybe Jenna as well. Is can we actually standardize things realistically? You know, given that the hardware we use is 3 years old, and we know it cannot be assumed to be stable. Like a lot of the Raspberry Pi or their camera is often discontinued. So in all likelihood, things will break. So maybe we can assume that we cannot reach hardware stability. What do you think?
I agree. I don't think we can. Like, we're already seeing with the AMI system, parts are being discontinued before we can even find a solution to get a new part. So it's really difficult. But I think in terms of, say, for example, maybe the light source, I would say, is one of the important things that we could standardize, because the power of that light and the type of light that we use it makes a really big difference, we found. So I think there's some bits that as a community maybe we could standardize, but other things are just not going to be possible.
Do you want to jump in, Andy?
Yeah, so I mean, I was thinking just on that note, how nice it would be to you know, have the data side standardized, and how much that would clarify things, especially from like the designer side. Like if I know, like, okay, we're all gonna have a bunch of different weird things. This thing takes photos every minute. This thing's out here for 10 months or something like that. But if at the end of the day, we all just have like, hey, we got a 2-dimensional image of moths from a fixed perspective, and this is the place it needs to go to. I think that could be a nice way to kind of unify and simplify a lot of like the design and science work that people are doing. So, you know, if I'm talking with some of my friends who work at like Wikipedia and do iNaturalist stuff right now, just about like, hey, like what format, what metadata, you know, what can we like dump all these things into as a community, and at least have that side standardized. I think that'd be a great target to have.
Absolutely. Mariana's got her hand up.
Yeah, something I've been thinking about is the sampling schedule. And with the first monitor, we had it taking pictures at 3 points in the night. And earlier in the night, we got a bunch of moths, and then later on, we had mostly mosquitoes. So this is something that's going to vary, I think, also with the particular biome where this is happening, because of the temperature that's throughout the night. but we could get very different metrics depending on the time of the night when we are sampling.
Yeah, definitely. And it ties in quite nicely actually with Christina's question about the fact that we can only monitor a specific area. Hopefully it's a somehow standardized area of a screen that we're looking at, and individuals are going to come in and out of that area. Ultimately, we want to get some understanding of how many insects are out there. But if if a single insect comes and goes, then there's this double counting issue. I think this is an issue that is prevalent in most ecological sampling, even traditional sampling, but it becomes very explicit in this case. And of course, the scheduling is going to affect how that works. So yeah, I think maybe if anyone's got any thoughts about how to handle this, individuals appearing, disappearing, or leaving the screen. Dennis, maybe you wanted to jump in on that as well. I think that's definitely an open question in this area. Jenny, you've got your hand up. You can segue from that, or
No, I can answer that. So there is the AI algorithm that's developed by the Rolnick lab, that does have a tracker on it, so it can track the moths as they're moving across the screen, so that it stops the double counting issue there. But then what it doesn't do is account for if the moth goes away and then comes back again. We don't know if the same moth is coming back 10 times a night, and it might depend on it might be species based as well. But I don't see a way around solving that. We've never managed to solve that with traditional sampling, like point counts with birds. You know, we don't know if we're recording one bird in 1 minute and then it goes and comes back again. It's unless you can identify the individual, we're always going to have that issue and need to move to measures of relative abundance rather than sort of direct abundance. So I don't see an issue around double counting fully, to be honest. I think it's always going to be quite difficult.
yeah, Christina's added about essentially if you have the time points of capture being more separated, then you have a smaller chance, presumably, of double counting. But then on the other hand, I think something that's really useful for this double counting issue, or for approximating the actual abundance of insects, is that occasionally you do get lucky and you have two individuals of the same species on the screen at the same time. And so if you capture that image, then you can be sure you have at least two, and that is a very informative image. So if you reduce your kind of sampling intensity, then you also reduce your chance of capturing that image which has two individuals or more of the same species, which is a very informative image.
Yeah, I would definitely agree with that. One thing that we've noticed is if you reduce the sampling intensity too much, we lose massive amounts of data. So when we've had the motion on, we collect everything, and then when we go to snapshots every 10 minutes, the amount of information we lose is massive. So we currently think that if we don't have motion turned on, what we need is snapshots. In the tropics, we need snapshots every 30 seconds, and in sort of more temperate environments, we probably need them every minute, between a minute and 5 minutes. So I think there's a trade-off there between your sampling intensity and yeah, you're going to lose a lot of data. But I think with camera trapping, they have like a 30-minute window, I think, of if it returns 30 minutes later, then it's a different individual, and it's perhaps one that's just they haven't managed to solve, and I don't know if it's solvable.
Yeah. So on the scheduling point, of course, we have scheduling of how often we take pictures, but we also have scheduling of our illumination. And so I think it's something that all of our presenters might have information on how often or continuously the sheet is illuminated, and whether that can or should be varied. I don't know if we could do a round of what our illumination schedules generally are. Should we start with you, Jacob?
Sure, yeah. So yeah, we've just been illuminating continuously. Both our illuminating white light and our UV light is on all the time over the course of the session. But we definitely expect different schedules to influence things. So I'm wondering if people have tried other approaches.
Andy, do you want to go next?
Yeah, totally. So also just hitting up, seconding this question, because this is something I'm very interested in, and there's all kinds of details I don't know. Like for instance, what's like the temporal resolution that like moths need? If I turn the big light on for like 5 minutes, is that enough to lure some in, or do they need to like warm up to it? It needs to be on for at least 30 minutes or something. And this is a big challenge for us because we basically like if let's say we only have 2 hours that we can sample the whole night. Is it better to like turn it on from like midnight to 2 a.m.? Or is it better to turn it on in like 10-minute increments from like 7 p.m. to 6 a.m.? Does anybody have any feelings about that kind of thing? That'd be lovely to know.
Yeah, I guess that's the interesting thing is mostly at this stage it is feelings. There are a few data on these kinds of things, and actually the automated system gives a perfect platform to collect data on this stuff as well. Mariana, what was your scheduling?
Yeah, so we've been thinking a lot about this because we want to balance attracting the moths, but then light pollution. We want to minimize light pollution. So what we did the first year was turning the light on, and then after an hour taking the picture. And then we took pictures every 10 minutes during the hour, and I was hoping to see accumulation curves and getting to a plateau, but we're not there. Something I noticed is that the landing surface area affects how fast the moths accumulate there. If you have a smaller area, it takes longer for them to accumulate than in a larger area. So some of the tests we want to do next summer is testing how long, so what's the shortest amount of time we can have the light on that we still get a good sampling. And if we vary the landing surface area, would that make it more efficient? And by more efficient, I mean shorter time the light on and more moths in the trap.
And then Jenna, did you have something to contribute on the AMI scheduling?
Yeah, so for us, we don't keep it on every night, just because we don't want animals, especially bats, to become habituated to the light. We don't want the bats coming down and feeding on the moth species. And we also, one really important thing is you don't want what we call a bird breakfast in the morning. So it's really good to turn these systems off, sort of a good little bit of time before sunrise, so that it gives the moths a chance to disperse. And yeah, so we have done some work on bats and the light source, and we haven't found any issues. But is that because we've only been recording 1 day in 3? If it was on every night, would we find an issue for that? So I think that's important. And then one thing we did find, we'll have more data being published on this in the coming months, but on an initial review, different species are active during different points of the night. So it does seem that there's a big peak of species. It's not that they're active all night. It will be different peaks for different individuals, and this could be clumped based on genus. I'm not sure, but I think in terms of your question, Andy, I think if you only have it on at one point, then you might miss a lot of species. So I think it is definitely good to spread that out as much as you can.
So we've got some really interesting questions coming in from Simon and Sarah about the kind of context dependence of the methods. So in Simon's case, open grassland versus dense forest, which I know from reading about light trapping in general, that being in dense forest can lead to a higher recapture rate than being in grassland. But does that apply for these methods as well? And also modifications for urban environments such as parks and cities. I don't know if any of these have been deployed in those environments. So any comments on that front?
Well, we want to try different habitat types, but we're not there yet. We need more of them. And for urban sites, We do some mothing on campus that's more urban than the field station, and you get a lot of competition with urban lights. So you just get fewer moths. And I guess for urban sites, you would also need some sort of a way of securing the equipment so it doesn't get vandalized or stolen.
Yeah, I think the issue of assessing the performance of a given sampling method as opposed to assessing the assessing the habitat itself in terms of what abundance or diversity of insects is that you don't really know at the point of capture. So the way this has been investigated in the past is you have a box of moths that you release at the site where your trap is, and you do that in different environments and see what the return of the trap is. It's extremely intensive exercise. Yeah, so it might be quite difficult to really get to the bottom of it. Yeah, so Andy, do you think the difference is due to the deep forest having higher or lower abundance and diversity of moths, or do you think it's due to the attractiveness of the trap, so to speak?
I don't know. I think it's just like it's still both like, rains here just like it rains over there and stuff like that. But just I think just even having like, closer, like if I go to like some forest that's kind of away from some like lights that we have in the city of Gamboa, we don't get as many, or we get different types of things as well, versus like if I go down Pipeline Road a little bit and it's just further away from other lights, it's like not only like the amounts are different, but like, I'll see way more like swarms of ants doing a nuptial flight closer in the big city of Gamboa. So just, yeah, even just, not very far distance, similar ecosystems, but, big differences is all. Oh no, I accidentally hit the recording button. I hope I didn't screw up anything.
I don't think so. It seems to still be recording. Jana, do you want to jump in?
Yeah, it's more to back up Mariana's point that we've really struggled with urban environments because of the light sources. So we recently had one of the systems at Chelsea Flower Show, and there was so many moths around the lights around the show, but then absolutely nothing on our system. So if there is any external light sources, I think it does make it really, really difficult. And also there's a lot of issues with the public, with people who live nearby. We've had our systems on lots of different farms across the UK, and we've had some of the farmers complaining that the light is so bright that it's like disturbing them at night. So I think we need to be careful where we put them so we don't upset people.
And I think we're coming close to the end of the session. So I thought I'd maybe just round up and say thank you so much all for coming. Go on, Andy, I'll give you a quick.
Awesome. Okay, I just had a quick question of something else I want to know. Has anybody dealt with physical elevation off the ground changes? Like how high up above the ground is your thing? And bonus, has anybody just tossed one in the canopy? Because that'd be really cool.
Yeah, I know that canopy moth traps do exist. I've never used one myself, but yeah, people have done that. They've designed traps that you can, hang them off of a branch. Yeah, if anyone knows any studies, stick them in the chat. Yeah, thank you everyone for coming, and please keep an eye out for the next webinar. I don't know if Quentin, if you want to stick you put the email in there again. So if you want to be updated about computational entomology events, please send an email to that address, and you're sure to be informed. And maybe a final thank you to the GINP, the Danish funding, and to the Research Coordination Network And yeah, thanks to all the speakers, Andy, Mariana, Jacob, and Jenna. Thank you so much.
Yeah, and thanks to you, Jamie, who spent a lot of effort organizing this first webinar. This takes more work than it looks.
It's great fun getting all these different methods into a big room and talk together.
All right. Bye, everyone. See you soon, hopefully.
Bye.
Thanks so much for organizing
Thank
this. This
you.
is great.
Yeah, thank you.
Thanks all. Bye.
Yeah.
Bye.